by the Neurobiology Institute. I am Aleph Prieto. I am new faculty in the Institute and have the honor to be the host of this session. Today, the keynote speaker is Dr. Karl Cotman of the University of California, Irvine. Dr. Cotman is a professor of neurology, and he is the founding director of the Institute for Memory Impairments and Neurological Disorders at UCI. Overall, his research is focused on identifying and testing intervention to reduce the rate of cognitive decline and promote successful brain aging. The research led by Dr. Cotman has been fundamental for our current understanding of brain dysfunction in aging and AD. For example, his group first discovered that beta amyloid damaged neurons and their axon and induced apoptosis-like mechanisms, including caspase activation. More recently, his work has been pivotal for developing intervention to promote successful aging, such as exercise. In fact, he was the first to discover that the exercise increases BDNF levels in the brain. Dr. Cotman has an outstanding record of publications, having more than 700 peer-reviewed scientific articles and over 120,000 citations. These numbers reflect the impact of Dr. Cotman's discovery. Finally, on top of his direct contributions to science, Dr. Cotman has empowered our field by the work of dozens of brilliant scientists trained by him. The list of successful scientists mentored by Dr. Cotman is large. Just to mention one example, the current president of the Society for Neurosciences, Dr. Oswald Stewart, is a former PhD student of Dr. Cotman. Thanks, Carl, for accepting our invitation, and please join me welcome Dr. Cotman. Thanks.
Well, thank you. I'm glad to be here to help you participate in the celebration.
You can go ahead, Carl, with your presentation.
Okay. Yeah. You know, I guess we're always looking for ways to improve the quality of life. And the dominant force in the field has really been pharmaceuticals. But, you know, it's kind of interesting that, you know, lifestyles haven't been as prominent. So what I'm going to talk about is focused on exercise and how it promotes healthy brain aging. And the key player appears to be BDNF. We discovered that first, and it's been replicated by over 250 different groups. Exercise drives healthy brain gene expression and counteracts age-related decline in humans. And so we've established that now. And with age, though, gene expression can be repressed. And we have a new clinical, and finally, then we have a new clinical trial ongoing to evaluate the action of exercise on cognition in humans with mild impairment, mild memory impairment. So I'm going to be telling you some of the key features of this story. You know, the biggest risk factor for Alzheimer's disease is just simple age. And you can see how fast the curve moves up from 75 to essentially 85. And so there's almost a 2.5-fold increase over that period of time. And as the population ages, it's been more and more critical that we manage to preserve cognitive function for these elderly individuals. So we need new treatments to maintain cognitive function as we age. And, you know, the startling news is there's been no FDA approved drugs since 2003 when the Namenda cholinesterase inhibitor was approved. And this is despite over 400 clinical trials. So it's a startling problem for the field and one in which, you know, we really need to get a solution to. On the other hand, lifestyles has emerged as effective strategies, and these include diet, exercise, cognitive and social activity. Probably the dominant one that has evolved and the more powerful one that has been studied is exercise, partly because it's simple to deliver, it can be quantified, and it's possible to have a parallel study in animals and in people. So the highlights are exciting, and I'll share that with you. So what are the potential modifiable risk factors for cognitive decline? And, you know, we see that, and you hear on the news all the time about diabetes and hypertension and obesity, which has become more and more of a problem as many older individuals have become sedentary. Smoking is also a problem, depression, but you know, so is cognitive inactivity and physical inactivity. So what's number one? In the US, it's actually physical inactivity. This was an exciting review and on the various risk factors done by Barnes and Yaffe in 2011, and it's held up pretty well. So we do see depression as being a second runner-up. Smoking is down in the US, but higher worldwide. So the risk factors, you know, physical inactivity can be addressed. And we figure if we could get people more active, we would actually be able to reduce the rate of cognitive decline considerably. And, you know, so it's been said that sitting is the new smoking. And unfortunately, we don't have a Surgeon General that's pushing the idea of exercise, just like that was previously done with smoking. So what are recent studies telling us? One area that's of interest, of course, is to see whether exercise improves capacity for learning and memory. And for that, the initial studies were essentially a radial arm maze, where the animals learn to essentially navigate a maze that's in water where there's various alleys, and they have to figure out where the food bait is in which alley and remember it. And what happens is that they will go and learn over a series of trials to learn where the target is and avoid the other mistakes. And it's really quite impressive. The rodents are probably better at running these kind of mazes than I am in finding my car in the parking lot all the time, especially because I have a small white car and there's a lot of small white cars. So anyway, exercise also facilitates consolidation. And one of the widely used tasks that we and others have used is object location memory. In this task, you put two objects in an open field in a particular place, wait 24 hours, and then test them again, giving them 5 minutes to select the object that is moved. And they will, to an astonishing degree, pay more attention to the object that's been moved. And so that gives you a measure called the discrimination index. And that is robust. And what we found is that if you give them exercise, exercise will take a sub-threshold score on the task and make it significant so it improves the discrimination. And it's really been replicated by many, many people. We uncovered it with a student of mine, Carly Inkelkoffer, in 2004, and it's held up very well. So exercise enables subthreshold learning. That's kind of nice because, you know, it's always when you're working, you want to have command of some of the issues that are really more subtle. And if you've, and with exercise, it would predict that you'd be better at some of the subthreshold learning things and can bring it up to the threshold and then keep advancing. So it increases efficiency. So exercise builds brain health. So what's the status of exercise interventions in humans? I gave you a couple examples of animal models, but let's talk a little bit about humans. The first real study that came out in 2008 was by Lautenschlager and published in JAMA. And what was amazing is that it's really took so long to essentially just do a study of a clinical trial in older people for what exercise did to cognitive performance. And so the first study was essentially 119 subjects with a subjective complaint of memory. They were randomly assigned to a home-based physical intervention or usual care for 19 months. The average increase in physical activity per week was only about 2 hours. So it's not like 2 hours a day or 4 or 5 hours a day. There was a benefit with just simply about 2 hours. And that improved a measure called the ADAS-Cog, which is a test that's widely used for pharmaceuticals. And the 1.3-fold improvement in performance with exercise is very similar to the initial improvement that was seen with the cholinesterase inhibitors. So it's really quite amazing. Subsequently, after Lautenschlager, there was a series of papers led largely by Laura Baker and co-workers, where she studied people with MCI and found executive function was particularly improved, you know, and so who doesn't want more executive function? However, the studies are reasonably small, and the results are somewhat variable. So as I'll show in the later part of the talk, that we need additional studies, and those are being done now. So some of the epidemiological data, though, is really quite astonishing. I saw this a few years ago, and here it is that there was a study published in neurology by Harder and group showing that physically fit women are 90% less likely to develop dementia. And this was 191 women followed longitudinally for 44 years and with different levels of physical activity. And only 5% of those with high fitness develop dementia. Astonishing. That's much under the US average conversion rate, which can get up to about 30 to 40% in the elder in the years. And in contrast, 25% of the moderately fit and 32% of the low fitness group develop dementia. So it's really a example of, you know, what persistent fitness can do and what and such remarkable benefits. So this is not an isolated paper. There's others too that back this up. So how does exercise affect the brain? You know, you just saw a glowing example of how it affects the brain. And, you know, it turns out that a neurotrophic factor or a growth factor that supports the growth and survival of neurons is increased. And this particular molecule is called brain-derived neurotrophic factor. It was initially discovered by Eve Barty and Hans Turninen group, and has turned out to be the really growth factor that's key to brain function. And particularly, it targets the glutamate cells, and some of the other ones, like nerve growth factor, do not. So brain-derived neurotrophic factor is basically like feeding the neurons essentially healthy nutrients, almost like a molecular fertilizer. And so, it keeps the neurons strong, healthy, growing, and able to resist to minor trauma, etc. So, brain-derived neurotrophic factor. After I discovered this and published it, some individuals, and I were at a ski meeting, and one of the guys comes up beside me and at the top of a hill and says, "Let's go get our BDNF levels up." So, people can think about this and have a lot of fun with it. Okay, so exercise does increase BDNF levels. And, when I first started this, I thought that maybe the levels would go up in the motor system the most, like the cerebellum or motor cortex. But it turns out that the biggest increase is in the hippocampus. And this shows the hippocampus here and how it lights up with exercise. And this is the area that serves a lot of cognitive functions. And so that's probably the locale where exercise increases the capacity to learn and remember. And it's linear related, but after it gets to a certain period, it doesn't keep going, of course. It does plateau out and will eventually go down, but the brain will maintain some of its health and benefits, probably due to structural change and just a healthier brain all the way around, kind of almost to the age of a more youthful brain. Okay, so how do we then prove that exercise is necessary for cognitive function? Well, the way you can do that is inactivate the BDNF. And there's a construct called a silencing RNA, or an siRNA, that if you give the animals and inject an siRNA with exercise, it blocks the exercise benefits. Here you see the gain in retention, but if BDNF levels are blocked, it's no longer able to remember. The animal is no longer able to remember. And that particular treatment not only blocks retention, but it also reduces the level of the BDNF. A very powerful molecular technique. Okay, so what is it that controls brain aging? That's a big question, and one that is really challenging. We know that the brain does age, and we just need to find out what it is. Now, normally what happens with age is sometimes the brain shrinks, especially in people that are not particularly healthy or cognitively impaired. So, but one of the things I was struck with is that gene expression might be a candidate, because when in all the studies I've done, I was puzzled by the fact that degeneration was not occurring as rapidly with age as the impairment. So there was a disconnect between the impairment and the change in the brain structure. So this led to the idea that maybe gene expression can be repressed, and so that would mean that the genes are sitting there but been inactivated. And there is a mechanism that can occur by, which is histone methylation. Histones are part of the gene expression system, and when they get acetylated, they can become activated, but when they're methylated, they generally are repressed. And so the question is, which is a very significant one, is does the epigenetic repression of gene transcription cause age-related neuronal dysfunction? Yeah, maybe. It's an interesting idea to think about, and one in which we addressed. And moreover, if it's repressed, can it be reversed, which is even more critical. So, there's a series of epigenetic repressors, and an important mark is this H3K9 trimethylation, which is an increase in the methylation at the lysine residue, where it gets trimethylated, and that turns out to repress gene expression. So is there a role for this H3K9 trimethylation in cognitive aging? Well, I was sort of interested in this, but had no idea how to test the idea. And this is one of these examples of where a university function and dinner can turn out to be really valuable. We were at a faculty dinner to celebrate people that had won the UCI medal, and I had won it one year, and another colleague that I knew pretty well, Larry Overman, had won it. And he's basically an organic chemist, and a really excellent one that is known worldwide for his capacity to develop complex novel molecules and synthetic pathways to essentially make them in a simple fashion. He just envisions it somehow, can watch the molecules moving around and then create a product. So he developed this inhibitor of the trimethylation called ETP69. It's a aromatic compound that's related to the piperazine class, and it inhibits the histone methyltransferase that drives the trimethylation, and that histone methyltransferase is called SUV39H1, and that's where the ETP will target it. So in this dinner, I got to talking to Larry and asked him, sort of what's new and what are you working on right now? And he was telling me, well, we're working on some compounds that inhibit histone methylation, essentially for cancer research, and to treat skin cancers. And I said, well, that's really interesting. I wonder if I could, have to think about that, because it might be interesting for our work. Anyway, I mentioned it to one of my postdocs, and she initially was pretty skeptical, that this would be worth her while. but I went and got some of the compound and from Larry. Had a nice visit in the process, of course, and came back and gave it to her and suggested that she try it and see if it improved learning. And nothing happened for a couple of weeks. Asked her again, nothing happened. And then finally, she comes to me one day and she says, "It worked. Wow, it's amazing." Says, "I finally got tired of you listening to you and decided it was easier to try it. So I gave it a shot, and by golly, it works." So this opened up a whole new area of research for us and a way to intervene at the epigenetic level to essentially control, to release the brakes on gene expression. So here's the aging actually turns out to increase. Just natural aging increases this H3 trimethylation in the hippocampus, not in the cerebellum, interestingly enough. This is a non-cognitive area, and although it's got some cognitive functions, but this one is generally not as plastic. And in the hippocampus, with age, there was an increase in this trimethylation, consistent with the idea that the animals do show a cognitive impairment as they get older. So that was pretty exciting. And then when that became known in the group, one of the other investigators, Li-Chi Tong, decided to look at aged neuronal cultures, because if we could get the system into culture, we could do more with it. And so he literally did an aging study in pure neuron cultures. And the advantage of that design is that we don't have the connective tissue, the vessel system to deal with, or glial cells in general. We can, if we get an effect of the H3 trimethylation, then we can know it's neuron, that we can study it in neurons. And so what happened is, with aging these cultures for essentially 7 days, 15 days, and 21 days, you get an increase in the trimethylation. And it turns out that this doesn't seem like it's very old, but the cultures in isolation age faster because they're under more stress, I suspect, and others do too. So there was an increase in the naturally in trimethylation with age. Now the ETP turns out to be quite selective to just different forms. And this shows the various enzymes that Larry and his group looked at. And, you know, because I got worried that if we don't have something that's really selective, it won't be meaningful and we'll not be able to get it published as well. So this target enzyme was essentially the top of the list, and other candidate molecules like SET8 and others are not affected by the ETP69. So there's a selective area and it's reduced in rodent hippocampus. Now, what is ETP69 able to do? Well, there's with age, mice, lose the capacity for object location memory, and if they're put into given two objects, like I showed it earlier, there's they can remember the area, remember the object that was moved. And if we gave ETP69 either pre or post the older animals were improved, whereas the controls were low. So the ETP69 is effective in improving their ability to recall discrimination and perform the discrimination. So they remember which is the new object and which is the old. And these can be really subtle differences, and it's amazing how the animals were able to do it. So the idea that it's be able to accomplish with a post-injection means that it's not affecting the learning process, it's affecting the consolidation. So that's important to essentially narrow down the mechanism. And so it seems like it's really acting on the at the synaptic level on what's called consolidation of learning. And it does change the structure of the brain and causes a growth of thin and spines. So the spines are where the synapses come in. This is the locus of long-term potentiation. So the spines can essentially be stimulated to grow by ETP, particularly the thin ones, which are the ones that are believed to be most plastic. So this is consistent with the idea that ETP is affecting synaptic plasticity right directly at the synapse level. And that defines the mechanism better. And so we began to wonder, maybe this is really a serious compound that is really efficacious. And so there's a transgenic model. The transgenic mice are ones that have had their genes manipulated so that they can make an Alzheimer's-like pathology. So you put in one of the molecules that is able to make beta amyloid, the amyloid precursor protein, and you put that into the brain and make it transgenic. And the 5XFAD mice, which is one of the more severe pathology developers, can't do the discrimination. But if they're given ETP, lo and behold, they can remember the discrimination. And these are very impaired by only a few months, only by 5 to 7 months. And so there's a real reversal of the impairment, and they're able to discriminate these things. So, okay. So what are the molecular targets of ETP? Well, we know it does increase BDNF. We showed that in some parallel studies. But what about other targets? We suspect that it activates CREB2, and so it gets to one of the main transcription factors involved in learning called CREB, and that is probably only just one of them. We haven't really fully explored the mechanisms down at the transcription level, and also what genes then does it really express. This is ongoing work, and we don't have real answers that I'm ready to talk about yet, but it's ongoing. Okay. Now, there's, I showed you some pharmaceutical strategies with the ETP69 that is able to reverse impairment, but we need, the ETP69 has not gone through toxicology studies yet to be used in humans. So we need a way to reverse the decline in learning and memory that occurs with age and Alzheimer's disease. So for this, our group, led by Aleph Nieto, Pedro Prieto, tried a novel approach, which is to see if we could study synaptic plasticity right directly at the synapse. And synaptosomes can be isolated and used as a novel approach. So Aleph Pietro did a series of studies, which I was really excited about because, you know, I did develop some of the methods for synaptosomal methodology and was familiar with them. It's not commonly used now, but it's coming back again. So you can then use synaptosomes to study LTP as a novel approach to study LTP directly at the synapse with the absence of glial and cell-cell interactions. And I call this the impatient approach because you can also do lots of samples at once by just studying synaptosomes, and it's a darn sight faster than single animal experiments to try to do LTP on each animal, where the most you could do is 4 to 6 samples per day. Whereas with synaptosomal LTP, you can do 40 to even more per day. So examined LTP and its impairment with age and Alzheimer's disease. And we wanted to determine if LTP can be restored in aging and in Alzheimer's synapses. Big order, but an exciting challenge. So here's the picture of a synaptosome. You can see the postsynaptic density, or the actual synapse itself, and the presynaptic ending with the synaptic vesicles that release neurotransmitter and cause the activation of the spine and its depolarization. So then this is a technique that we used. And here is the methodology. Notice that here's Aleph published the paper in 2017, and other studies as well. And so you isolate synaptosomes, stimulate them chemically, because you can't electrically stimulate a synaptosome without a lot of trouble. And so you then stimulate with a chemical mixture that simulates the electrical pulse and depolarizes and activates the NMDA receptor, which is the key signal. And then you look at whether they're potentiated by the surface expression of the glutamate receptor A1, which is localized postsynaptically. And when it's not stimulated, its N-terminal is internal, but to the cytoplasm. But when it's stimulated, it becomes externalized due to exocytosis. And then you can detect GLU-A1 extracellular. And you can then pick that up by flow cytometry to pick up the labeled things. To make sure that you've got actually intact particles, we used a presynaptic marker, Nerexin 1. So we then looked at the ones that were attacked synapses by showing the coexistence of Nerexin and glutamate AMPA receptor, GLU-A1. So what happens when you, we first wanted to show that the system really was well behaved and was essentially able to get LTP with chemical stimulation. And here's the expression levels of the glutamate receptor and the Nerexin. And you can see that with basal, there's a certain number of particles, but with stimulation, there's a increase in the number of particles that have been potentiated. This compares to electrical stimulation of slices. We did a rather necessary experiment to electrically stimulate a brain slice and then isolate synaptosomes and show that you could get an increase in expression with electrical stimulation. And so that validated the approach to show that it was like electrical physiology. So, and then using that, we could ask some questions that are really otherwise unapproachable and study long-term potentiation in the human brain. And I have to give Aleph a lot of credit for this, because these are high-risk experiments, and you might do these experiments and get nothing out of it. But he had ventured in, and the data turned out to be really quite revealing compared to controls, which were younger synaptosomes. The AD ones had an impairment in the potentiated response when you gave them chemical LTP. So you get a increase with in the controls, but in AD synapses, they're not potentiated. So they're basically, non-plastic, which kind of people assume, but this was the first demonstration of that. Okay, and it was age, sex matched, and PMA, post-mortem interval matched. And this was in human AD synapses. So what happens is really actually remarkable. Indeed, you can get potentiation and LTP in the human synaptosome if you coach it a little bit with a phosphodiesterase inhibitor to raise the cyclic AMP levels and GMP levels. So then you, because the system apparently in isolation from human synapses has lost the essential levels. And so if you booster that with a phosphodiesterase inhibitor to bring the levels higher, then you can actually get the potentiation. This means that you might be able to essentially treat some Alzheimer's patients with analogs of these drugs that are usable in humans and see an effect and improvement. This has not been done yet, but might be feasible, indeed feasible. So a nice breakthrough that, you could see something. And you couldn't do this without looking at postmortem tissue. We were fortunate that in the institute where I work and where Aleph worked, we have a good brain repository, which I established years ago when I ran the whole program. And, this access to postmortem tissue allows you to go from animal studies to human studies. And it makes it more exciting because you really want to do things relevant to humans, of course. Okay, so anyway, we're moving along here. And, these people are feeling their phosphodiesterase and their cyclic A and cyclic GMP levels, and also in bolstering them indeed. So what about physical activity? What does it do to gene expression in the human brain? Our specific functional categories impaired. So with age, there's two major areas that the age attacks in terms of the function of the brain and its structure. One is a decline in mitochondrial function, and the other is a synaptic decline. And these are some of the references where that concept has been published. So Might physical activity counteract these age and AD related declines at the transcription level? It's kind of a big order, but you know, if you don't try it, you don't find out either. One of the things that I try and do myself is try things that I don't think will actually work, but that are logically rigorous, and see if you get a new discovery and a breakthrough. And indeed, that's really what happens and what happened. So the study design is to look at physical activity and gene expression patterns. And where the heck are you going to get brain tissue that where people know how much activity they have dealt with that the patient had before they passed away, and then get access to the brain? Well, you know, I sort of, the person has probably one of the best brain banks is at Rush Medical Center, David Bennett and his co-workers with Buckman, Aaron Buckman, etc. And so I thought, well, maybe they would have it. I met David at a meeting when they actually had in-person meetings, and asked, told David what I was going to do, I'd like to try, and asked him if he had any samples where they had some idea of the physical activity, either through a survey or something like that. And he says, oh, I can do better than that. He says, I've actually got Actical monitor data on individuals. Well, Actical is like a pedometer. And so he actually had quantitative data on how much physical activity these individuals had. So we could then look at those which were low activity versus high activity and see what happens to the gene expression patterns. This was an experiment I could hardly wait to see the result from. So here's the activity patterns. We had low and high activity patterns, and we compared the low to the high, you know, and tried to emphasize the big differences. And we then took those samples and did microarray analysis on the human hippocampus, and we used a physical activity data set and an aging AD data set to compare. And I identified genes that are significant in either aging or AD with physical activity. So this sets the experiment up, and the tissues are really valuable because, you know, these are not just well-characterized older people that have had cognitive testing, and these are cognitively intact individuals. And also they had the activity levels. So we compared the directions of change. I'm not going to go through the whole chart. I'm going to give you the bottom line on it. It's been published, Neurobiology of Aging, last 2019, if people want to look at it. But basically what happened is really pretty clean. I would have been happy with, frankly, a few genes that responded, but what we found is 95% of the genes showed the opposite pattern of change. So if they went down with age, they went up with exercise. And basically the dominant thing that happens with aging is that gene expression is decreased. So, we got an increased expression though with physical activity. So, and then we can look at the genes that were enriched for specific functions, and found out that the genes that go up are mitochondria and synapses, just like the literature would predict is what goes down with age and is needed. there were also some, the transcription machinery that makes proteins that were affected. So we then looked at what happens to the mitochondria, and here's the genes in the electron transport chain. So you can see this is the electron transport chain, that every complex in the electron transport chain, from the initial complex 1 to essentially 5, and as some genes that were increased, and indicating that the mitochondria is being rejuvenated with physical activity. Really remarkable therapeutic impact. And indeed, it was related to the dose. The individuals at high activity had more gene expression than the individuals with low activity. And this essentially tended to reverse the aging AD decline, whereas with physical activity, you recover it. Boy, what do you just perfect, you know? So the fact that I thought a few things might be improved, we've really found that quite a number of them improved. So to summarize, and then I've got a little bit on a clinical trial that we're doing to tell you about. Physical activity is a major risk factor for cognitive decline. And so sitting is the new smoking, so to speak. Human microarray analysis showed dramatic decline in overall gene expression with age in healthy cognitive non-demented individuals, and further decline in AD. Physical activity counteracts the age and AD related gene expression changes. And then learning can be studied in synaptosomes, and PDE inhibitors restore LTP even in AD synaptosomes. So those were the main conclusions of that. Exercise does indeed build brain health, like I said. And so now I want to just close in giving you a couple minutes of a large-scale exercise study that's being done in people with mild cognitive impairment. And, you know, working on animal models is rewarding, but really wanted to translate our findings on exercise to humans. And so I was thinking about this, and I was part of a clinical trials group, and it proposed this, that we do a study of exercise in humans. And there was a lot of enthusiasm for it. What finally evolved, to make the story short, is that UCI and the NIA have partnered with YUSA. We also brought in Wake Forest, because Wake Forest had a particular investigator, Laura Baker, who is extremely seasoned in doing exercise trials, and we partnered with her, and she's been a sheer pleasure to work with. And so what we've done is use the YMCAs to be a site to serve it, because we wanted some place where it was accessible and you know, doable, you know, in a community setting. And so we then did EXERT. We called it EXERT. EXERT is the exercise trial. Kind of a simple thing. Laura thought of that, and darn clever. I designed the X and stuff, and fussing around, you know, one doodle, etc. And so what is EXERT? Well, it's essentially, you do screening, and then we have essentially a 12-month exercise period, which can be extended without supervision for longer, and do various testing, including CSF and brain imaging. We do their conditioning effects, and then carry it out for another 6 months to essentially 18 months. So, you know, that study is ongoing. We have essentially, we had a goal of enrolling 300. We enrolled 296, and just completed that several months ago. So the study is fully enrolled, and some of the people that started initially are now completing their exercise. You know, we've had incredible retention. It's been remarkable how once people start on this, they love it and stay with it. And had some testimonies of how, you know, one particular woman who had considered herself sedentary got into this program, and she testifies how she enjoys it, and she thinks she's sharper as a function of it, and more active and energized, and interacting better with her friends and community. So the study, the initial methods paper has been published, and we will then be going out with the rest of it soon. So this takes it from animals to people, and hopefully with the YMCAs. The YMCAs are now coming back from COVID. I would say that we had the COVID pause at 6 months. So that was essentially a setback. But we have the first 6 months of data that's just clean. And we're now bringing back individuals. So it'll be kind of almost a field study with distractions and other things. And so we will find out how people are doing with this. Okay, with that, I'd like to acknowledge some of the key people. Nicole Birchow, Chris Butler, Shika Sinja, Aleph, who's now moving to the University of Mexico, and of course, and various other collaborators that we've worked with that have been invaluable and partnered with things. This is only the recent collaborators. I've not gone through the other ones. So thank you. An exciting time with many opportunities yet before us. We're opening up a window where indeed lifestyles can make a huge difference. And, you know, I guess the old adage is true, use it or lose it. You know, keep moving. Okay, thank you so much. And that's all, folks.
Thank you, Carl. Thank you. Very nice presentation. We already have questions. This is the QA section. And Dr. Fernando Peña has a question. Maybe you want to make him by yourself, Fernando? I think yeah, you have an open, yeah. Okay, Fernando.
Yeah.
Go ahead.
So thank you very much for your presentation. I have some questions. The first one is there any equivalence between sedentary lab rats and some level of activity in humans?
I'm sorry, I couldn't understand the question.
Yeah, I'm just wondering if there is any equivalence between sedentary lab rats with some kind of activity in humans.
Yeah, the sedentary individuals, you know, 40% of the US population is what you'd call sedentary. They get less than 2 hours of activity per week. You know,
I think,
but what about rat labs?
Well, the lab rats, I mean, there's different ways in which animals are housed at baseline, and that's a problem that's not standardized in the field yet. So when animals live in small cages, they have restricted activity. When they get in bigger cages or together with others, you know, then more activity, et cetera. It's a whole way of affecting the baseline, very big time.
Okay, and the other question is, I saw that there is a difference between BDNF expression between CA3 and CA1. Do you have an explanation why those two regions respond differently?
No, but it's generally, that's what people do find. So that's it. And incidentally, hello there. Nice to hear from you. Yeah, the CA3 and CA1 is a big mystery. And Dentate is generally where more of the action has recently been with the BDNF levels. So it's initial relay between the cortex and the hippocampus proper.
Okay, finally, I have a final question. Is there any secondary effect of this H3K9 inhibition?
Secondary effects?
Yeah.
Side effects, yeah.
We haven't done a lot of injections, but in culture, it's essentially non-toxic. It's really quite remarkable. You can give, you know, ETP and raise the levels, you know, and don't see any toxicity. But I'm sure there's toxicity, you know, once you get into it. We, in the periphery, they've done toxicology studies on fibroblasts, and there's essentially no toxicology within the normal working range that's needed for therapeutics.
Okay, thank you very much. It was a very nice talk.
No, thank you. Nice to hear from you.
Yeah, you too.
Dr. Fernando Barrios, do you want to make your question?
Hi, yes, of course. Can you listen to me?
Yes.
Okay. Well, first of all, thank you very much, Dr. Cotman. It was a very nice, very interesting talk. And I was just wondering if, well, obviously this is activity, physical activity, but nevertheless, is there any specific activity that is recommended at older age? That's one of the questions. This compound with that question is that if once there is an inset, I mean, there is a cognitive impairment or whatever, has been well established that with exercise, with physical activity, we can reverse the effect or not in humans.
Yeah, good question. The types of activity range from walking to essentially intense aerobic activity. And I think it's kind of like the amount versus the intensity to some degree, and there's a trade-off. So we don't know, essentially, that's why the actigraph studies are key, because the total activity seems to be the most important from what we can tell so far.
Mm. And is there, besides walking and whatever is this general activity, is being measured and you know, recovery of cognitive, you know, cognitive impairment? Is there a recovery with exercise?
Yes, you do see improvements.
Okay.
It's amazing.
Oh, well.
It's a BDNF, I think. But in humans, of course, we can't test that.
Well, yeah, okay. Well, thank you very much.
You're welcome. Thank you.
Now we have a former student of Dr. Mago, Dr. Gina Kirarte. Yeah, Gina Kirarte knows pretty well your work and has a question. Gina,
Sure.
please.
Do you hear me?
Yes.
Oh, okay. Great talk, Dr. Cotman. It was really nice to see how exercise improves memory. So
Thank you.
in almost all the studies that you present, you test the animals 24 hours later, which is long-term memory. So I wonder if BDNF also facilitates short-term memory.
Well, it depends on what you mean by short term. You know,
well, I'm thinking in, you know, like the test that that a lot of people do, which is you don't remember what you have for breakfast, but you remember what you did 1 year ago.
right. Well, you know, BDNF does improve short-term memory in animal studies and even in human studies, and physical activity will do that. Whether it's BDNF or not, I don't know. You know, the effects of BDNF are quite remarkable, and they're very robust. And I think one of the next windows of opportunity is going to be to try and combine some promising pharmaceuticals with physical activity to see possible synergisms, because each are going at slightly different mechanisms and should be synergistic.
Okay, thank you. I wish you were here in Querétaro with us. So
It would be nice. This didn't happen.
yeah.
Dear old virus.
Thank you.
You're welcome. I wish I was there too.
We have another question by Dr. Teresa Morales, the director of the Institute. So please, Tere, please go ahead.
Okay. Thank you very much for your presentation. I really enjoyed your talk, and I appreciate that you were willing to give us the talk this way. So my question is—
nice to meet you, incidentally.
Yes. Is there an influence of diet in the results of the study by Harder in from Sweden, and also in your results on mitochondrial function? Is there an effect of diet? I mean this because in Sweden, most of the diet is based on fish. So maybe that could have an effect on the results.
Yeah, we did a whole diet study with aged dogs and beagles, actually. And what we were able to show is that a diet rich in, all things leafy greens and, essentially other food substances that were supplemented are really good for the animals. And in fact, so good that Hill's Pet Foods copied it and now sells it. So I guess, it's preferred. But diet's definitely a big factor. And in human studies, various investigators have done human studies. But boy, I'll tell you, it's hard to control a human study for diet because people don't stick to them. And, it's been done, but most people don't find big effects.
So, okay, thank you very much.
Well, thank you.
I think we have time for another question. Diego Ramirez Gonzalez, do you want to make your question yourself? Ask. Okay, I can ask the question of Diego Ramirez Gonzalez is that if do different types of exercise have different effects on cognitive decline, or if are they all the same?
Oh, good question. Actually, in animals, that's not been studied in detail. So I can't give you an animal answer, but the impression is with with humans, the decline has been how long after exercise is exercise still effective, and that gets into a story we developed called molecular memory, where there's a molecular memory for the exercise that lasts about 3 to 5 days after it's stopped, and you still retain the benefits, and then they eventually go away, but it's primed then to be reactivated with high efficiency. So that part, I think there are other benefits that haven't been studied yet, but it's a very promising area of research.
Okay, thanks. Rebecca Hernandez have a question. It is the following. Would the levels of BDNF in the hippocampus of aged people maintain similar levels to the young age with the daily exercise?
Maybe certain isoforms, but I doubt it if the whole thing, it's not been done. The postmortem samples for humans is very limited. Particularly Yeah, I just don't know. I'm sorry.
Okay, Dr. Carmen Clapp, do you want to ask your question yourself? Carmen Clapp? Okay, I can just read the question.
Okay.
Carmen Clapp is asking, I would like to hear your comments regarding whether short-term memory versus long-term memory is affected in old age.
Yeah, short-term memory is definitely affected more with age. You know, people really can't, especially retrieval. I mean, the big problem seems to be retrieval, if you know, but short-term memory is really compromised. You know, it's the idea even finding the car in the parking lot sometimes, you know, gets challenging for older people. You see them wandering around the parking lot looking for their car. So spatial learning, which is a hippocampal task. And hippocampus does shrink with a little bit with age, but that some investigators have shown that in humans, a half a year of physical activity can increase the volume of the hippocampus. Work done by Kirk Erickson and group. Very exciting to think that you can actually increase the size of the hippocampus with exercise.
Thank you.
Another question is, I like this question. Is there a specific cell type responsible for the exercise-induced BDNF release?
Pyramidal cells, particularly.
Okay.
The interneurons, I don't think have been studied in much detail, but they're not believed to be the source. You know, you can get what cells are secreting it by the mRNA levels, and you know, the pyramidal cells have a good level of BDNF in all of its exons.
Okay, Ivan Montiel, do you want to ask your question yourself? You are connected,
please.
I can see. Okay.
Yes, please. Nice talk, doctor. I have a question, and first at all, thank you for your presentation. The first question is, could physical activity benefit any kind of type of memory?
Procedural learning is not particularly benefited, you know, such as a motor skill, but that's just simple practice, and that's probably the least effective by other exercise. Now you can, you know, design studies. Strength training, curiously, does not seem to translate to improve cognition for the studies that have been done.
Okay, and the second question is, these findings that you presented are intended for people with memory impairments, but could they be extended to people who don't have these necessarily the treatments in memory?
Say that again. So can it be extended to?
And these findings that you presented is for people with memory impairments. And the question is, these findings that you are presenting could be extended to people who don't have this cognitive impairment?
I think so, because, you know, the gene array stuff I showed you from humans is actually on cognitively normal individuals. So the gene expression that related to the amount of actical activity was really normal individuals. I probably didn't make that very clear.
And finally, the last year we have the presence of the Marcelo Wood. And Marcelo Wood presented results of how the epigenome integrates information and reshapes the synapse. And the question is, do histone modifications and other epigenetic modifications represents a molecular substrate of memory? This is very clear. In that way, how these findings can change the concept of engram?
Good question. Actually, Marcelo and our group are collaborating now on some studies on the molecular memory part. Yeah, I think the whole histone, I mean, I showed you a little bit of the methylation story, you know, which is a histone modification. And so I feel confident that the exercise is going to have a whole story unfold at the histone acetylation and methylations end. So, good question.
Okay, thank you so much for your patience and good talk. Thank you.
My pleasure. Thank you for the question.
I think this was the last question. Carl, on behalf of the members of the Institute of Neurobiology at the Universidad Nacional Autónoma de Mexico, I'd like to thank you again and just congrats for your great and as usual, nice talk.
Well, thank you. And it was a pleasure to interact with you all.
Okay, well, I think you are finishing this presentation. Thanks, and see you tomorrow for the next activities from the academic journey of the Institute of Neurobiology. Thanks.
Okay, you're welcome. Bye-bye.
Bye.